All right. You might be done with the electron transfer chain, but we're not done with the mitochondria quite yet because our next topic heavily involves them, and that is apoptotic signaling. Our learning goals today are going to be to compare the intrinsic and extrinsic pathways of apoptosis, and we will also be examining where these two pathways overlap. That will involve recognizing instances of overlap or crosstalk within these apoptotic signaling pathways. Lastly, we will be identifying the 8 common characteristics of cellular signaling that are observed largely in apoptotic signaling itself. The molecule of the moment should look familiar. It's cytochrome C. And cytochrome C, if you recall, shuttles electrons from complex 3 to complex 4 in the electron transfer chain. It is not membrane bound, it's kind of membrane associated. It's a soluble protein that exists within the mitochondria's intermembrane space. However, when the cell experiences stress, the mitochondria will release cytochrome C into the cytosol, and once it's out there, it will begin to trigger the death cascade. Therefore, this is interesting, cytochrome C is a protein that keeps the cell alive or kills it. Depends on where it is in the cell at a given time. So the questions for you are twofold. Number one, remind me what the term is for when a protein performs more than one function. We've seen this a few times. The second is a little bit more of a thinker. I want you to tell me what is the benefit of utilizing cytochrome C in two completely different pathways, one being a keep me alive pathway and the other being a kill me pathway. We've spent several lectures discussing the biochemistry of life, so it's high time we featured the biochemistry of death. Death at the cellular level generally comes in two flavors, one being necrosis, and the other is apoptosis, point of order. You may have noticed by now that I deliberately call it apoptosis, and that's because that's the way it's supposed to be pronounced. The second P in apoptosis is silent. You don't say it. You don't say pterodactyl, you say pterodactyl. And you don't say apoptosis either. It's misleading. The cell doesn't pop. That's really more of what happens in necrosis anyway. And it's just wrong. And I don't know why it hasn't been corrected yet. So anyway, it's important moving forward that you understand the general features of these two outcomes. We'll first circle in onto necrosis. This is unplanned, premature cell death. It's when a cell becomes so horrifically damaged that it swells and then eventually ruptures, pops, and spills its contents and non-functional organelles into the surroundings. And this is bad because when everything on the inside of the cell is suddenly on the outside of the cell, the immune system begins to freak out. This is going to trigger a horrifically inflammatory reaction that's going to result in more and more unplanned cell death. So this pathological feedback loop is going to propagate all of this tissue damage as a necrotic wave. You can see in this image over here that the frostbite of this hiker happened at the tip of their toes, but oh, the damage and blistering is starting to work its way down their tissues, starting to turn yellow and then gray and then black. So the triggers for necrosis are, generally speaking, some kind of a trauma. It can be as simple as a knife wound, a severe contusion, any sorts of those things. Like I mentioned, frostbite, not good for any tissue. Animal venom, cobra toxin is a very dangerous necrotizing poison, I believe. And also some severe infections. And when I say severe, I don't mean a really bad case of the flu. I mean things like necrotizing fasciitis, aka flesh-eating bacteria. That is going to cause necrosis in your infected tissues. So what are the ways to fix this? Well, you've only got two options, really. You can debride the tissue, scrape away the dead parts until the living parts are okay. And that can be quite a bit of scraping away and cutting away. If that's not enough, you may have to amputate the affected digit or limb before the damage propagates out and takes over your whole body. So necrosis is not a good thing. Apoptosis, on the other hand, is programmed cell death. Instead of the cell popping and bursting open, apoptotic cells shrink and safely disperse their functioning organelles into little vesicles known as apoptotic bodies or blebs. These blebs don't create mass hysteria. They tend to be anti-inflammatory or outright physiological, and those blebs can get scavenged and recycled by nearby cells like phagocytes. So therefore, unlike necrosis, apoptosis is frankly protective sometimes. When do we see this occur? Fetal development, for starters, has lots of deliberate apoptosis in it. You may have learned that babies have webbing between their fingers and toes, at least in the early days of development. What happens is around here, apoptosis is going to create notches that will eat away at the webbed fingers. This greatly increases the dexterity of the person after they're born. That's a physiological application of good programmed cell death. And even after development, apoptosis occurs all the time. When that child is born and is exposed to the horrors of the real world, like viral infection, mutation, radiation, afflicted cells can take themselves out before they become problematic or cancerous. It's better that a few cells die for the sake of the entire organism. That's the philosophy here of apoptosis at that stage. But make no mistake, apoptosis is not something the cell wants to do frivolously. So my goal now is to outline the ways apoptosis is initiated and regulated through cell signaling. There are two ways in theory to initiate apoptosis: the intrinsic pathway and the extrinsic pathway. My goal is to just discuss these in broad strokes for now. We will be going through these both in more detail in the slides to come. But for now, I'd like you to know that the intrinsic pathway, as the name sort of suggests, it's triggered by internal sensors. things like DNA damage, unfolded proteins, oxidative stress, so on and so forth. All of these are signals that the cell isn't keeping it together. And if the cell can't keep it together, it can't be trusted to live and create future generations. So the mitochondria are going to sense this, and they will ultimately commit the cell to a death pathway, which you see outlined in the bottom right over here. We'll talk more about that later, though. The extrinsic pathway, however, has an apparently less convoluted mechanism of signaling, or so you may think. The extrinsic pathway is when an external signal called a death ligand comes over and binds a receptor on the surface of the cell that is being instructed to die. And once the receptor binds that signal, it generates a series of reactions and protein reconfigurations throughout the inside of the cell that ultimately lead to the activation of caspases. And if you look at both of these graphics, caspase activation, whether it's the intrinsic or extrinsic pathway, converges onto these caspase 3 and caspase 7. Caspase is not an acronym, a portmanteau of the word cysteine aspartate protease. That right there is a clue that we have some sort of catalytic dyad in a protease that is working to chop up critical proteins inside the cell. It is a somewhat promiscuous protease that loves to munch at the cytoskeleton in particular, the thing holding the internal infrastructure together. So you can imagine how once the cytoskeleton is gone or weakened, how things can fall apart rather quickly. I think you're ready now to take a look underneath the hood of the intrinsic pathway of apoptosis. And it begins with something known as the basal state. The basal state is when a cell is hunky-dory. Everything's happy. We're doing our job just fine. Cells are constantly expressing regulatory proteins that just sit around in the background, like BCL2. These serve as apoptosis suppressors. And that makes sense. If we're healthy, we don't want to die, right? So let's have these green flags fly about the cytosol and maybe even neutralize the red flags that we're also making at the same time, like BAC and BAX. These proteins are pro-apoptotic. And so the cell is kind of caught in this delicate balance where the amount of BCL2s needs to exceed the presence of BAC and BAX so they can neutralize all of them and maybe even have few to spare and ensure that the healthy cell doesn't accidentally go down the road of apoptosis. But we're not foolish for having BAC and BAX in the foreground here because we need to be ready to release them if and when disaster strikes. And what does disaster look like? Well, we learned that hypoxia threatens our ATP reserves by jamming up oxidative phosphorylation. This also will generate reactive oxygen species, which destroys the cell and the membranes. Extreme temperature, like freeze-thaw cycles or heat waves, can really screw up protein structure, mess up protein folding. And let's not forget our mutagens, viruses, radiation, carcinogenic compounds. All of these things can alter or physically damage the DNA running the show here. This is where a protein called P53 comes to the rescue. P53 is, in my opinion, the coolest, most important protein in cancer biology. It's lovingly known as the guardian of the genome. Its job is to suppress cancer at all costs. If there is a little bit of stress going on, it will actually rally DNA repair mechanisms and also protein repair mechanisms if there's a misfolding crisis. If the cell is really stressed, P53 will arrest the cell cycle, and it says no more dividing until we're back to homeostasis. And if that doesn't happen in a certain time frame, well, P53 makes the tough call and triggers apoptosis. When the damage is too deep, simultaneously, P53 will suppress BCL transcription and upregulate Bax transcription. It will also bind and refold BAC and Bax into a more potent configuration. So we can see them giving it a super upgrade. BCL2 is gone. Now we have mega Bax and mega BAC, and there's no apoptotic suppressors around to stop them. They will now swim over and bind to the mitochondrion and initiates the following. Those BAC and Bax proteins oligomerize into pores that literally punch holes into the mitochondria that are just about the size of cytochrome C. Cytochrome C is therefore no longer trapped within the intermembrane space. Cytochrome C escapes, diffuses, really, and all hell is now about to break loose. This process, where the cytochrome C molecules, our molecule of the moment, escape, that is the committed step of the intrinsic pathway. Once you cross that boundary, there's no going back. Another word for this process is MOMP, which is short for mitochondrial outer membrane permeabilization. After permeabilization, when cytochrome C leaks into the cytosol, it will bind and reconfigure and activate a dormant cytosolic protein complex called APAF1, apoptosis protease activating factor 1. When cytochrome C binds it, check out the cartoon over here, it's going to cause this very complicated structure to unfurl like so. With a little help from ATP, we can unwind this multicolored, complicated set of domains to create an exposed sticky region down there, shown in orange, And that's going to recruit and bind 6 other activated APAFs. So altogether, when this process is done, we are going to get a cool cyclic heptamer protein snowflake beast called the apoptosome. And look at how cool this quaternary structure is. I mean, seriously, this is easy to miss because this tiny red dot there, and there is cytochrome C. That tiny protein in these zones is what orchestrates all of this material coming together. Pretty sophisticated, right? Now, the main job of the apoptosome is to activate an inhibitor caspase called procaspase 9. It already exists in the cell on call as the monomeric zymogen. Remember that word, zymogen? It's an inactive enzyme, usually with a masking sequence clogging its active site. Well, the apoptosome has very specific protease activity to it. It's able to cut that masking sequence and convert procaspase 9, inactive, into dimerized, active caspase 9. And now the job of these initiator caspases is to prepare the cell for cell death. They actually don't carry out the deed themselves, but what I do want to highlight here is the similarity to the blood clotting cascade. It's so often we propagate signals through a chain of proteases, activating more proteases. It's a great way to amplify a signal to get a fast response. The caspases responsible for the final phase, demolition phase, of apoptosis are the executioner caspases 3 and 7. The initiator caspase 9, its job is to remove their respecting masking sequences blocking the executioner caspases, shown over here. So once the initiator caspases activate the executioner caspases, now they are ready to do some killing. However, the design of the binding pocket of caspases 3 and 7 ensures that they go about the killing gently. They are not going to blindly snip up every single protein in the cell. Instead, they target various cytoskeletal and nuclear structural proteins. And when they do that, the cell begins to systematically lose its structure. So there are indeed a few hundred substrates for these executioner caspases. I don't expect you to know them all, but in a word, I would like to share the highlights, like actin. Executioner caspases love to chop up actin, and actin's job is to provide the cell with rigidity. Various cellular adhesins are also fair game for the caspases. These proteins allow the cell to stick to its neighbors, and also the nuclear proteins gotta go, the ones holding the nuclear envelope intact. Next on the hit list, though, is proteins like BCL2 and P53. Isn't that interesting? All the proteins I talked about so far were structural. BCL2 and P53 are functional. Why do you think nature evolved the executioners to recognize these guys? And many, many others similar to it? I'd like you to pause and maybe flip back a few slides and try to work that one out for yourselves. Mwahahaha. With the cytoskeleton degrading faster by the millisecond, the cell hurtles towards the final stage of apoptosis, which is the Bleb cycle, the end result of either the intrinsic and extrinsic pathways. Remember how the execution or caspases weaken the actin cortex of the cytoskeleton? Well, it's going to do that unevenly across the membrane's surface. See how over here the actin is thinnest? That creates a weak point in the membrane. And remember, the inside of the cell has a lot of hydrostatic pressure. It's exerting an external force on its environment. And its ability to contain that force is now compromised with the actin being degraded. And so it's kind of like a cellular hernia. This is my analogy. I don't believe anyone else would know what you're talking about if you said this. But with the actin gone, water pressure is going to seek to relieve itself by rushing into the part of the membrane that is, well, exposed. And that's going to cause a bleb to form. And the blebbing zones are going to get bigger and bigger and bigger as more and more of the actin gets degraded away. So this next part seems a little bit counterintuitive, but the cell wants this to happen. The cell wants blebs. These are the safe, protected vesicles that distribute the contents of the cell's will without creating mass hysteria. And so, even though I said that the caspases are chopping away at the actin, within the environment of the growing bleb, we will see a reversal of that. We will see actin formation, which stabilizes and pinches off the bleb. That pinching off portion is actually attributed more so to myosin, and that process requires ATP. So once you do that and you form a bleb, then the whole process is going to continue. And this process happens until you have no more membrane left, no more cell left. All you have are apoptotic bodies, which are readily consumed by phagocytes. Let's now rewind and consider the other apoptotic pathway, the extrinsic pathway. It might appear simpler than the intrinsic one to the untrained eye. I don't know. We'll see how that turns out. I'll just remind you that in this approach, we are triggering apoptosis externally through the application of a death ligand. Something like TNF-alpha, which is very important, showing you it over there. TNF-alpha is something that would be released by, let's say, a white blood cell as a way of instructing the cell receiving said factor to undergo apoptosis. I will now draw to your attention that the TNFA molecule is a homotrimer. Its quaternary structure consists of 3 identical primary sequences bound together in a triangle fashion, and that is going to bind not 1, not 2, but 3 death receptors on the surface of the target cell, the receiving cell. And as that trimeric death ligand drags these humongous TNFR1 receptors together, at the same time, we are positioning the attached cytoplasmic domains to also come together in proximity. The receptors and their associated cytoplasmic domains don't really do anything until they come together in pairs of 3. And when they do, these regions over here, called death domains, will combine and change conformation. These new activated conformations now has binding affinity for certain adapter proteins. These are like literal adapters that can connect various protein tools in the cytoplasm, each triggering a different pathway. It's like a domino effect. The death domains bind an adapter called TRAD, and then TRAD binds FAD, and then FAD ultimately binds and cleaves and activates an initiator caspase. Procaspase number 8. It is the formation of the disc, aka the death-inducing signaling complex, that constitutes the committed step for the extrinsic pathway of apoptosis. Because once we have that, we now have activated initiator caspase. And what do initiator caspases do? They activate the executioner caspases, procaspase 3, procaspase 7, the same ones that we saw at the end of the intrinsic pathway, which means you already know what happens next. Cytoskeletal demolition, then blebbing, then formal death. So it's time for a brief history lesson. The major death ligand for the extrinsic pathway, TNF, that stands for tumor necrosis factor. And its name is really misleading. First of all, if you're sharp, you may be wondering, why is it called tumor necrosis factor? We just learned that TNF triggers apoptosis, not necrosis. Why is it called necrosis factor then? Well, that's because TNF was discovered not before the term apoptosis was invented, but before the term got popular and caught on with the scientific community. So TNF became a big deal as apoptosis was starting to become a big deal, and the inaccurate name unfortunately just stuck around. But perhaps more attention-grabbing is the first word in that acronym, tumor necrosis factor. That makes it sound like this signaler can kill cancer. Well, back in the 70s, scientists were already investigating a very controversial treatment for cancer called Coley's toxins. A scientist by the name of William Coley noted that sometimes cancer patients went into remission when they experienced a severe, life-threatening infection. So he and his followers hypothesized the following. What if we injected some nasty bacterial antigens termed Coley's toxins? And what if we injected them into the site of the tumor? Would that provoke a strong immune response around the cancer cells and hopefully kill it in the process, all without actually making the patient sicker with a secondary or opportunistic infection? It's a similar philosophy to crafting vaccines when you think about it. So they tried this in rats, and they noticed that, well, if we graft a tumor into a mouse and inject the tumor with Coley's toxins, indeed, the tumors shrank and died. Except additional experimentation revealed that the toxins themselves weren't poisoning the tumor. Instead, the toxins rallied the immune system to produce a certain protein factor, and then that factor is what helped kill the tumors. And of course, the factor that they isolated was, you guessed it, tumor necrosis factor, hence the name. And oh boy, scientists were delighted. Did we just discover a breakthrough cure for cancer, and one that was hiding inside our own bodies the whole time? Well, you can imagine the fervor over the next few years trying to isolate it, study it, purify it, scale it up, all with the intent to inject TNF into cancer patients as a Hail Mary cure. Well, the clinical trials for exogenous TNF therapy for treating cancer began in the 80s, not very long after the discovery of TNF itself, and I'll say in a nutshell that it was an epic failure. The drug did not kill or shrink tumors. It in fact made the patients dangerously sick. It gave them severe fevers, immense pain, hypotension, all classic symptoms of shock. So what gives? Why did the TNF injection kill the tumors that we grafted in mice, but it made the clinical trial subjects severely ill? And what's even worse is phase 1 clinical trials, these are toxicity studies. Patients that enroll with them are given subclinical doses, an amount that's expected not to cure the cancer. They're just trying to find out if a little bit is dangerous or not. And it turns out it most certainly was. The reason why the TNF-alpha clinical trials failed so disastrously was because the scientists did not anticipate crosstalk. Crosstalk is an incredibly important dimension of cell signaling that was just frankly not understood at the time. What do I mean by crosstalk? Well, let's revisit this earlier scheme where we break down the extrinsic and intrinsic pathways. Remember how this graphic sort of neatly silos them into two separate beasts that will eventually converge onto the common ground of executioner caspase 3-7 activation? Well, what if I told you that there were convergences that preceded this step? What if I told you that BCL2, the anti-apoptotic protein, not only inhibits back and backs, but it also inhibits initiator caspase 8 of the extrinsic pathway? This is likely why those advanced tumors weren't dying in the human trials that featured the recombinant TNF. Cancer can, over time and in certain tumors, ramp up BCL2 as a protection mechanism. BCL, by the way, stands for B-cell lymphoma. Guess where that protein was first discovered in alarming amounts in the first place. Now that's just one instance of crosstalk. What about the opposite? Well, if CASP8 is successfully recruited into the TNFR disc complex, CASP8 can actually activate BID. And BID can neutralize BCL2 while directly activating BAC and BACs, which are pro-apoptotic. Hell, even the executioner caspases are able to directly activate BAC and BACs as well, which accelerates the process even faster once it's begun. And there's plenty more instances featuring these pathways and others, but I think you get the point here. Like metabolism, signaling features feedback loops and feed-forward loops and activation and inhibition all working together. So that previous slide explains why adding so-called tumor necrosis factor to a tumor doesn't necessarily result in the death of said tumor. But it also doesn't explain why the patients that enrolled in that ill-fated clinical trial got so sick from the experimental TNF therapy. And the reason for that is ultimately the modular nature of cell signaling itself. Okay? One ligand doesn't evoke just one cellular response. Don't get me wrong. Yes, the death ligand TNF alpha binds the death receptor TNFR, which recruits its death domains to bind the death adapter, and that can lead to the activation of apoptotic signaling. However, TRAD over here is a fork in the road. It is a highly modular adapter protein with the capacity to bind other non-death adapters, such as TRAF2, which is a signaling pathway that leads to survival. And thanks to a healthy presence of anti-apoptotic proteins like BCL2, which get in the way of procaspase 8, this progression of TNF alpha to TRAD to TRAF2 is actually the default state. I know it seems kind of counterintuitive because it certainly sounds like it, that TNF alpha, the death ligand, in most instances prompts survival. But I like to think of it this way. When a cell needs to die, it is going to be exposed to TNF alpha while it tamps down protective proteins like BCL2. The result of that is going to be cell death. When things are hunky-dory, the cell is going to have a baseline level of BCL2, which is going to keep in check any TNF that wanders in its direction. So instead of going down the death road, we're going to go down the survival road. And that's a good thing. We don't want accidental TNF wandering by and just triggering death in an otherwise healthy cell. But we still have the option to apoptose if the situation were to suddenly change. Now, let's imagine what could happen when we have a healthy or unhealthy amount of BCL2, and we have a large amount, in turn, of TNF. We see this happen in a number of inflammatory diseases and cancers, and the result of that is not just survival, but inflammation. We're not just going to survive, we're going to fight. And what does fighting look like? Well, that's eventually going to result in the activation of this transcription factor I've boxed over here called nuclear factor kappa B. It is going to upregulate several defense proteins, such as BCL2, That's an inhibitor to back and backs, and it's going to further promote survival. We don't want to die at this point, we want to fight. Further cementing that response, we will transcribe for XIAP. These are caspase inhibitors. Again, we're going to make apoptosis even more difficult to achieve. We are going to synthesize interleukins, leukocyte recruiters. If the cell is in trouble and is in defense mode, we're going to call over the infantry and try to get it to help us. And to help the infantry, we will also make pyrogens, aka fever inducers. These instruct the body to raise its average temperature, and it will also generate aches and pains and all the other sorts of unpleasant effects of just feeling sick and crummy. Nuclear factor kappa B will also make a bunch of collagenases. Collagenases are going to weaken the cell-cell contacts surrounding the affected cells, which is going to allow leukocytes to infiltrate the problematic area more easily by generating some wiggle room. And this is going to alter the transcription of dozens, if not hundreds, of other proteins, but I'll end this by noting that nuclear factor kappa B is also going to make more TNF alpha. So this entire affair is going to be a positive feedback loop. The cell will make the inflammation worse and worse and worse until the problem goes away or we die trying. This is precisely what happened in those clinical trials for TNF therapy. A lab-grown tumor grafted into a mouse has a different cellular chemistry than a tumor that has been growing and adapting and learning inside of the person for years. In time, cancer has the potential to teach itself to be anti-apoptotic, if not outright inflammatory, by really leaning into protective mechanisms like BCL2. If you grow a fake tumor in a petri dish over the course of a few weeks, it's not going to have the same level of robustness. That is ultimately why administering exogenous tumor necrosis factor was not a cure, and it in fact just made those poor people sicker. So I shared all of this history with you to teach you an important lesson, and that it's really easy to overlook signaling pathways because they're messy by design. And almost always, we tend to know less about a situation than we think we do. And boy, I can have an hour-long conversation just on that. So what can we distill from all of this other than that signaling is messy? I chose the TNF axis as our introduction to signaling biochemistry because it features numerous hallmarks and properties of cell signaling at large. And the 8 characteristics of signaling that you should be aware of are specificity, sensitivity, amplification, modularity, divergent, integration, adaptation, and localized responses. And what we will do now together is walk you through each of these characteristics and how they've applied to what we've just learned. The first items I'll highlight are specificity and sensitivity. Ligands are famous for binding their receptors with so-called high fidelity. This essentially means that the death receptor is only going to be triggered by its corresponding death ligands, like TNF-alpha. And this specificity originates from the geometry of each ligand matching the receptor's binding pocket exquisitely. To visualize that, I'll divert your attention to the cartoon on the left, where we have two ligands competing for a common receptor shown in red. We have L1 and L2. These are different ligands with different shapes. L1 is going to fit into that binding pocket perfectly. It's going to click in, induce a conformational change in the receptor, activate it, and bam, you get a response. L2 is free to collide with that receptor's binding pocket. It's just not going to stick, and it will therefore not trigger that response. So no significant interaction occurs. L2, however, is free to diffuse on over to its appropriate corresponding receptor and trigger that response. In short, life wouldn't work if ligand-receptor interactions weren't specific, because we'd be constantly misfiring the wrong responses to the wrong signals. It would be a disorganized mess. Now, sensitivity is also important because, well, it's not practical or wise to flood our blood with high concentrations of primary messengers. So the way we address this is by having receptors readily dock low concentrations of ligand, thanks to having very low constants of dissociation, aka a low KD. This is synonymous with having a very high constant of association, a Ka. And I'll remind you that the lower the Kd is for a binding event, the tighter the bind, the more equilibrium is going to favor the adducted state. Let's visualize this property with our friend TNF-alpha. In a healthy person, the blood or serum concentrations of TNF are approximately 0.6 picomolar. That is practically nothing. It's 1,000 times more dilute than nanomolar. And if you're curious, this is equivalent to 0.6 times 10 to the negative 12 molar. So it's really, really dilute. On the other hand, the constant of dissociation of TNF with its receptor is also quite low. Its Kd is equivalent to 1.76 nanomolar. So nanomolar quantities of TNF in our blood would, in theory, saturate half of the corresponding receptor. So I'm wondering, 0.6 picomolar, what kind of receptor saturation can we anticipate from that level of ligand? To answer that question, this equation down here will tell us what percent of receptors are bound, theta, at a given ligand concentration, L, assuming it has an affinity, Kd. So all we need to do is plug these numbers into this fairly simple fraction, and we can find out the theta value. I will warn you, though, that the units are different here. We have picomolar and nanomolar, and a picomole is 1,000 times more dilute than a nanomole. So I'm going to plug these values into this equation with one modification. I'm going to convert everything to picomolar, including the Kd. And I do that by moving the decimal place over 3 times. So it's going to be plus 1,760. And this is going to give us a very small number. I'm going to put this in my calculator now. And yeah, the theta value is equivalent to 0.00034 which is to say, if I move that decimal place over twice, .034% binding. Now, that doesn't seem like much, but that's all that it takes to have a healthy baseline activation. We are achieving this with next to no ligand, 0.6 picomolar. Let us now take a gander at the theoretical saturation for TNF's doomed clinical trials. I actually pulled up those old reports and calculated that a typical dose given to those patients was an infusion consisting of 340 micrograms of TNF-alpha isolates. If we multiply that by 10 to the 6, that will give us how many picograms were in that dose. I'm going to assume the molecular weight, based off my calculations, of TNF-alpha to be 51K Daltons per mole. Oops. Excuse me. Let's neaten that up a little bit. That means we have approximately 6,666 picomoles present. The average human adult contains 5 liters of blood. So if we divide these two numbers, we will get a final concentration of about, I'm going to write it over here, 133 picomolar of TNF-alpha, plus, I suppose, the 0.6 picomolar, which we can ignore. Let's put this back into that equation now. We're going to get a very different result. 1333 picomolar divided by 1333 picomolar plus 1760. That will equal to look at that. 0.43, aka 43% saturation. That is a very sensitive response. We took 340 micrograms of protein. That is less than 1/10 of the mass of a grain of sand. And by injecting that into a person, we've saturated half of all their receptors, give or take. If that doesn't convince you we're sensitive to these signals, I don't know what will. Next up, we have amplification and modularity. Both are highly featured in apoptotic signaling. Signal amplification is crucial because, like I just mentioned, there's not a whole lot of signal floating around in the bloodstream. Serum concentrations of TNF-alpha are next to nothing. So the best way we can utilize small quantities of ligand is to exploit amplification cascades. One death ligand is all it takes to activate multiple initiator caspates, and each casp8 is then going to activate many executioner casps 3s and 7s. The amplification seen over here is philosophically no different from the blood clotting cascade. By geometrically growing our signal, we can get fast response times. Blood clots in seconds, apoptosis in hours. So the key takeaways are one signal activates many initiators, and each initiator activates many executioners. Geometric amplification equals fast response. We also get a taste of modularity when we encountered the many adapter proteins associated with the TNF receptor. One set of adapters causes inflammation, the other causes death. And there's frankly many more that go beyond the scope of this lecture, much less this entire course. The key is a so-called singular death ligand is able to navigate all of these responses thanks to the modular nature of these adapter proteins. Binding and phosphorylation events can reversibly change adapter affinities to the receptor's cytoplasmic domain. And that is what ultimately dictates which adapter successfully binds. So, flexible cytoplasmic domains plus flexible adapters equals many possibilities. Integration and divergence are the hardest characteristics to grasp in my opinion, but this is why I shared that story about those ill-fated TNF clinical trials. Decades ago, for example, we didn't fully grasp how signaling pathways are integrated through crosstalk. Now we know that death ligand signaling is going to be in competition with survival signaling. And if you look at this graphic underneath my excellently drawn skull and crossbones and angel face, you're going to have two ligands, let's say TNF and a growth factor, both be competing against the receptors against the cell surface. Perhaps the TNF signal is ramping up the activity of some secondary messenger, whereas the growth factor is causing it to deplete. Well, which one wins? It's whichever one has more ligands pressing the buttons. So the net change in whatever stimulus is being inputted is going to dictate if we're going to swing to the death sides of things or swing to the survival side of signaling. If integration didn't happen, one wandering death ligand could theoretically kill every cell it bumps into because there's nothing counterbalancing it. Or likewise, if you have both flying around in the milieu, the cell might be constantly swinging back and forth between contradictory physiologies without actually accomplishing anything because it's just getting confused and overwhelmed. So in a word, I like to think of integration as navigating contradictory inputs wisely. As for divergent signaling, which is when a receptor gets activated and it generates many simultaneous responses, Well, I think glucagon is a great example of that. We learned about glucagon, right? Triggering its receptor activates protein kinase A. Protein kinase A eventually activates glycogen phosphorylase, so that means glycogen breakdown, while we also shut down PFK2, which is going to slow down glycolysis. So the way I like to think of divergent signaling is multitasking. To my knowledge, localized response and adaptation don't generally apply to apoptosis since that's a whole cell phenomenon, but I want you to think about chemotaxis instead. Chemotaxis is an excellent example on how a signal can be localized to microenvironments within the cell. So white blood cells move towards other cells that release interleukins as a cry for help. And they can do this because the white blood cell has interleukin sensor receptors all along its exterior. If this square cell is in need of help, it's going to have a concentration gradient of interleukins extending out from the center. The white blood cell does not have eyeballs, it does not have ears, it cannot see or hear its cries for help, but it can feel them. So it's going to chemotax in the direction of the highest concentration of interleukin. They do this because the receptors on the side of the cell closest to the cell in trouble are going to get triggered the most. So the secondary messengers generated are going to be localized in that cardinal direction. If this localization didn't happen, then that poor white blood cell would get pulled in all directions because the secondary messenger cascade would completely fill the interior of the cell and it wouldn't know which way is which. As for desensitization and adaptation, this doesn't exactly apply to the apoptotic context either. It's kind of all or nothing. But you do experience this on the daily, where a signal influences the sensitivity to the stimulus. Take a moment to, I don't know, flick your cheek repeatedly in the same spot. I'm doing it right now. The first one kind of hurt, but they eventually got less painful and just frankly more annoying. That is your mechanoreceptors desensitizing themselves to a painful stimulus. You know, if you're in public and you don't want people seeing you flick yourself for no reason, think about going nose blind to a perfume you're wearing, or the garlic you had with lunch that now everybody but you can smell. Bam. All of that is desensitization of receptors. So common examples are going nose blind, and hell, even drug dependency. Have you ever heard the saying that addicts are always chasing their first high? That's because every time they take said drug, the body becomes less and less sensitive to it because the receptor presentation gets tamped down in the presence of overwhelming signal. So we spent quite a bit of time unpacking the intricacies of apoptotic signaling, yet we barely scratched the surface. Yes, apoptosis is something we still don't completely understand, but death ligands and cytochrome C are just two of thousands of types of signalers that our bodies are attuned to. Lots of the signalers out there are chemical, all right? I want you to think of, I don't know, hormones and neurotransmitters, even other cells. These are all chemical entities that can influence the physiology of a given cell. But life also responds to non-chemical stimuli, the physical, like light and touch, all right? Pressure, even. Hell, the absence of oxygen, that's a stimulus. Without signaling, maintaining homeostasis is going to be impossible. Thanks to signaling, we can smell, we can taste, we can grow, we can develop, we can fight infection, we can do all these wondrous things. So that's why we will be circling back to this topic in the next lecture to more fully unpack all the different ways signals can be propagated throughout the cell.